So now we're going to turn to talk about what happens when you make molecules that are partly hydrophobic and partly hydrophilic. These molecules are called amphiphiles, and you see on the slide a structure of one. This zigzagging symbol here stands for rows of methylene groups. So these are CH2s that bond to neighboring CH2s. And finally, we have a CH3 group at the end of the chain, so terminated in a methyl group. So each of these vertices is one methylene group. This is a classic, very non-polarizable hydrophobic molecule. In essence, it's chicken fat. Notice that this molecule, though, now terminates in oxygens, which, as we know, have lone pairs that are charged. And indeed, you'll see at the end a Zwitter ionic group. Notice the phosphorus here. And then coupled via a couple of methylene groups and an oxygen to this group here, which is positive. So this has a large dipole moment. Once again, it's a Zwitter ion. So this region of the molecule here is hydrophilic, and this region here is hydrophobic. This means that when many of these are present in an aqueous solution, the hydrophobic interaction will tend to push this part, the chicken fat part, together, while having this part, the ionic part, in solution. They don't just self-assemble with themselves. When you get an oil stain on your shirt and put it in the laundry, you'll add, when you shake the detergent bottle, a molecule very much like this, with an oily part and a water-loving part. So this part will bond to the oil in the oil stain on your shirt, this part will pull it away into the water, and the result is a clean shirt. Such materials are capable of forming remarkable self-assembled structures. The simplest type is perhaps just this micelle, in which if I draw the hydrophilic group like this and the hydrophobic group like this, the hydrophilic groups just stay on the outside of the structure, and the hydrophobic parts sit on the inside to form a little spherical shell. This is a micelle. One can form, though, a lipid bilayer in which the hydrophobic groups associate like this, leaving hydrophilic groups on the outside. And this then can form a double-shelled structure with hydrophilic groups on the inside and the outside. So that water can be contained in here, this hydrophilic region, and water can hydrate the outside. This is a liposome. Double-walled structures like this are extremely important because, in fact, living cells consist of little bags that are themselves lipid bilayers with hydrophilic groups pointing on both the outside and inside, and hydrophobic groups filling in the inside. The evolution of the cell was perhaps one of the single most important accidents in chemistry, because this allowed distinct chemistry to go on the inside and material to be concentrated and accumulated in a micro-reaction vessel in a way that wouldn't happen in bulk solution. Finally, this slide shows what can happen if you form these materials on a flat surface, or if they interact in just the right way, they can form a stable bilayer which is flat. And this is just called a lipid bilayer. These structures will form spontaneously. This shows you a series of steps taken from a molecular dynamics simulation of a small block containing a random mixture initially of amphiphilic molecules and water molecules. And as time progresses, you can clearly see the phase segregation of the amphiphilic molecules as their hydrophobic regions, shown here by this browny color, tend to come together. And as time progresses further, you actually see a sheet beginning to be formed. Here's the sheet starting to curl around, and eventually we're beginning to form a liposome.